channel Clinical Biochemistry by Dr. P.K. Prabhakar. Today we are going to talk about DNA. Basic structure of DNA we will talk about. We know that DNA is the deoxyribonucleic acids, and this is present in almost all the cells. Wherever nucleus will be there, so DNA will be there. Are all the characters, all the properties depends on DNA. DNA is the normally decides are all the features. Either it will be internal features, or it will be external features. So all the characteristic features, physical properties, all the metabolic reactions depends on this DNA only. Our colors, our behavior, our different properties depends on DNA. So DNA we will see how it happens, and slowly will different lectures will explain how DNA decides our character. So this DNA, as we know, this DNA is the one of the component which is present in the cells and decides our characteristic features. This DNA is first discovered by F. Misters in 1869, and then after 80 means almost 100 years, 80 years, it has been explained that DNA is the material, DNA is the components which is the genetic material which decides our characteristic feature, which can be transferred from one generation to the other generations. So that will be explained by Avery, MacLeod and Macquarie in an experiment. So that experiment also we will discuss in the coming lectures. So now we will see DNA. This is a polymer, polymeric structures. So DNA is composed of different nucleotides. Not only DNA, in our cells basically there are two types of nucleic acids we are having. So this is nucleic acids which is present in our cells. Either it will be eukaryotic cell or it will be prokaryotic DNA will be there. So this nucleic acids we are having two types. It will be either DNA or it will be RNA. DNA is deoxyribonucleic acid. So this is deoxyribonucleic acid, and this is ribose nucleic acid. So later on we will see how DNA is different from the RNA. That also we will see. So now today we are going to talk about DNA. So both the nucleic acids, either it will be DNA or RNA, it is a polymer. So both are polymer of nucleotides. So DNA as well as RNA is composed of number of nucleotides joined together. So these nucleotides are one of the most important nitrogenous metabolites. This contains nitrogen. Number of nitrogen will be there. So normally nitrogen present in our cells in different forms. So all the nitrogen content of our cells we are going to divide into two different broad classes. It is called as proteinous nitrogen and it will be called as non-proteinous nitrogen. Proteinous nitrogen normally present in the amino acids and in the form of amino acids is present in the proteins. Non-proteinous nitrogen present in different places. It will be in the nucleotides, DNA. Non-proteinous nitrogen also present in the urea, ammonia. So those are different non-proteinous nitrogen sources. When we are talking about non-proteinous nitrogen sources, DNA is one of the important non-proteinous nitrogen source where nitrogen is present in our cells. So nucleotides are found primarily in the monomeric units, which gives you both DNA and RNA. So now, the monomer of DNA is called as, so this is polymer, its monomer we are calling nucleotides. Nucleotide. This nucleotides basically composed of three different components. So the first component of this one is the nitrogenous bases. Nitrogenous base. Second component of this one is ribose in case of RNA, and deoxyribose in case of DNA sugar. This is 5 carbon sugar which is present in DNA or RNA. Either it will be oxy or it will be deoxy. We will see when we are going to talk about sugars. That time we will see what is difference between ribose and deoxyribose. And third one is phosphoric acid. So these 3 components are present in the nucleotides. If you'll see this one, so nucleotide equal to nitrogenous base plus deoxy sugar plus phosphoric acid. When we are having this nitrogenous base and deoxy sugar, we are calling it nucleoside. So this is called as nucleoside and this is phosphoric acid. So in other terms, you can call it nucleotide equal to nucleoside plus phosphoric acid. So we'll see how this is going to form and how it occurs. Now first we'll see the nitrogenous bases. The first component of our DNA is nitrogenous bases. So those 3 things are joined together and gives you the nucleotides. So there are, if you see about nitrogenous bases. In our DNA, there are 2 types of nitrogenous bases are present. One is called as purine, other one is called as pyrimidine. You can remember it. The name among this purine and pyrimidine, purine is a small word, so its structure will be big, and pyrimidine is a bigger word, so its name structure will be small. So this is 6-membered structures. Out of these 6 members, 2 members will be nitrogen. So now you can see, you can remember it. This is This is 6-membered structures. In this case, we are numbering in anti-clockwise. So number 1, number 2, number 3, number 3, and in the purines. So the structure will be this is 1, 2, 3, 4, 5, 6, 7, 8, 9. So nitrogen is present in first position, third position, seventh position, and ninth positions. In case of pyrimidines, nitrogen is present at first position and third position. So this is the basic template of purine and pyrimidine. In our cells, we are having 2 types of purine is present. We are having, so I'll rub the structure and then we'll write it. If you'll see purine, we are having adenine, we are having guanine. If we'll talk about pyrimidine, we are having cytosine, thymine, and uracil. These are the 3 pyrimidines. We will see a structure of these 5 nitrogenous bases one by one. So adenine, the structure of name, you can remember the name. If you remember the name and you are remembering the basic template of the pyrimidine, purine and pyrimidine, you can write it automatically. So adenine is called as 6 amino purine. So first I will write the name and then we'll talk about the structures. Guanine is called as 2-amino 6-oxypurine. It is called as 2-amino 6-oxypurine. This is one member rings which is having only one rings. So the name of cytosine is 2-oxy 4-amino 2-oxy 4-amino-pyrimidine. The name of thymine is 2,4-dioxypyrimidine. Pyrimidine. 2,4-dioxy 5-methylpyrimidine. This is 2,4-dioxypyrimidine. Now we'll write the structure of one by one. So this one are basic template for purine. Now if you write adenine, so this is the basic template of purine, and if you write the adenine's structure, so it is 6-aminopurine. You can remember, you have seen the nomenclature when I have done the numbering. So this was number 1, number 2, number 3, 4, 5, 6. So if you put at here amino group, so this is our adenine. So this is number 1, number 2, 3, 4, 5, 6, 7, 8, 9. So if you are going to write 6-aminopurine, so this is our adenine. If you are going to write, remove this one, guanine is 2-amino 6-oxy. 6-oxy means this is 6-oxy, and at second place it is having NH2 group. So this is 2-amino 6-oxypurine. So this is our guanine. Similarly, if you are going to write about the pyrimidine structures, so basic template of pyrimidine was this is 1, 2, 3, 4, 5, 6. So if you are going to write about cytosine, it is 2-oxy. So at second place oxy, at fourth place amine, MH2 group, and pyrimidine. So this is our cytosine. Similarly, thymine will be 2,4-dioxy. So this is oxy. At fourth place I will put again oxy, and at fifth place we will have methyl group. So this is called as thymine. The difference between thymine and uracil is thymine is having this methyl group, whereas uracil does not have methyl group. So uracil is 2,4-dioxypyrimidine. So this is our uracil. If we have seen structures of this one, both purine and pyrimidines, so we have seen different groups, different molecules. There are some things you can remember it, which is coming in the as a MCQ questions, like difference between this one and this one is deamination of cytosine. If you remove the methyl group from this thymine, you will get uracil. So demethylation of thymine will give you uracil, whereas if you means you can interchange it one another by adding one group or second group. Now if we have seen the structures, in this one. Like I'll write this one and then basically you will understand it. I will write 1 purine, adenine I am going to write. So 6 amino I am going to write. This is R adenine. I am going to write thymine. Thymine will be, this is 1, 2, 4 dioxy methyl group. This is our thymine. If you see both these structures, there are different molecules, different groups will be attached there. Like here, oxygen is there, here ammonia is there, and some places nitrogen is also there. So you have heard about the hydrogen bonding, hydrogen bonds. Normally, in our case, there are three. If you see the periodic table, there are 3 different elements which is a strong electronegative elements in our cells, in our periodic table, that is fluorine, nitrogen and oxygen. These 3 are one of the strongest element in the periodic table, which is having the highest electronegativity. So whenever these 3 molecules, these 3 elements associated with hydrogen, either FH, NH2 or NH or OH will be there, and at a specific distance fluorine, oxygen and nitrogen will be there. So there is a hypothetically a kind of attractive force works there. So this attractive force is called as hydrogen bonding. So in case of hydrogen bonding, the hydrogen, so there is a attractive force between this hydrogen, which is associated with the electronegative elements, and the other electronegative elements. So this hydrogen, which is directly associated with this fluorine, nitrogen or oxygen, any of one, and a hypothetical bonds will be there with the other elements. So this fluoride, fluorine or nitrogen or oxygen is called as hydrogen donor, and these are called as hydrogen acceptors. So whenever a hydrogen bond has to form, there will be a hydrogen donor and hydrogen acceptors. Never hydrogen bonds will form between 2 donor, where OH and OH. Never 2 hydrogen bond will form between the 2 acceptors. So always for the hydrogen bonds we need 1 acceptor and 1 donor. Means if I have to form the hydrogen bonds, so because this does not have hydrogen bond, so this will work as a acceptor. So we can put a arrow towards this one, means it is a hydrogen acceptor. Similarly, this is having hydrogen, so it will always work as a hydrogen donor. So we will denote a arrow away from that one is hydrogen donor, and arrow towards a molecule we will call it hydrogen acceptors. So always hydrogen bonds can form between only this hydrogen donor, hydrogen acceptors and hydrogen donors. This can work as a hydrogen acceptor, this as a donor, this as because it is having hydrogen, so it can work as a hydrogen donor. This does not have any hydrogen, so it can work as a hydrogen acceptor. So never hydrogen bond forms between this one and this one, because both are acceptor only. So that we are going to see it. Sorry. Other than this one, these nucleotides does exist in different tautomeric forms. Tautomers are by the shifting of hydrogen. When we are hydrogen will be shifted from one element, one particular place in their structure to the other places. So that is called a tautomeric structure. So we are not going to remove hydrogen, we are not going to add hydrogen, we are just going to change the place of hydrogen in the same structure. So hydrogen is going to interchange their structure, their places. Like, so that is called like tautomeric structures. Like if you will write cytosine. this is your cytosine. So cytosine structure is 2-oxy. This is 2-oxy, and this is NH2. So this is amine, and this is nitrogen, which is hydrogen. So it can exist in another forms where one of the hydrogen from here will come here, and this hydrogen double bonds will shift here. So here we will have double bond like this one. So that is different types of tautomeric structure. So we are having that, we are going to see it. So almost all the nucleotides exist in two different tautomeric structures. So these tautomeric structures can be like ketoenol. If you will see it, we are having ketoenol forms like OH, C, H, this type of structure. This is in all. This can exist in these forms, keto forms. This is in all forms. It can exist in the form where this is called keto form. So we have not done anything. We have just this hydrogen from here, we have shifted it here. So hydrogen is changing one place from one place to other place. This is called as ketoenol tautomeric forms. So if you'll see our nucleotides, they exist in different type of forms. So this is one kind of structures, one kind of tautomeric structures. Other than this one, we are having another structures where almost same occurs, but in place of carbon we are having nitrogen. So here this is between tautomeric structure between carbon to carbon. Hydrogen is coming from carbon to carbon. So this is carbon to carbon. If we are having similar type of things will be there, but it will be from nitrogen to hydrogen will be transferred from nitrogen to carbon. I will show you the structures. So that is called as amide or amine, imidic forms. If same thing will be there, it is amide, amide, amide plus iminoforms, imidic forms, yeah, iminoforms. This is when it is going to occur in case of open chain structure. If same thing occurs in case of ring structures like we are having nucleotides, so there we are calling it lactam and lactim. So this is in open chain, but when it occurs in same nitrogen between carbon, it occurs, we are calling it lactam, lactim. I will show you the structures. So amide and amido first we will write, and then we will see lactam and lactim. Same, everything will be same. It is this is where nitrogen is having hydrogen. This can exist in the form of OH. This is in all. Similar to in all, it is keto forms. So this is difference between, in case of here the hydrogen is transferred from nitrogen, in case of enol it is hydrogen is transferring from the carbon. So that is the difference. So this is in open chain, we are calling it amide and iminic forms or amino forms. Iminic, you can call it better. If this occurs in a ring structures, it is called as lactam and lactim. So in our nucleotide structures, whatever nitrogenous bases we have seen, so they exist in lactam and lactim forms, like almost all the things like guanine, thymine, cytosine, they exist in lactam and lactim forms. What is the difference between, what is the significance of these tautomeric structures? This affect their hydrogen bond forming natures. So in one structures, 2 hydrogen bonds can form. In other structures, less hydrogen number bond can form. In other case, it will be more hydrogen bond can form. That we will see it. Now, so thymine can exist. Thymine's structure, if you see it, thymine was it was 5-methyl 2,4-dioxy. This was 2,4-dioxy 5-methyl. Now this is keto forms, because at both the places we are having keto group. Now this can exist. This is keto forms, you can call it, because it is in the ring structures, and hydrogen can transfer from, this is NH, hydrogen is going to transfer from nitrogen to carbon, so you can call it lactam forms. It is also called lactam form because it is ring structures. So now it can also exist in another structures. These are the structures. What will happen? This hydrogen from here, it is going to transfer to this one. So, and this double bond will be shifting here. So, if you see this place, so this is the keto group, and this is in and all. So this is called keto. This is, you can call it enol, because more specifically it is called as lactam forms. This is lactam, this is lactate. Similarly, guanine also exists in keto or lactam forms, and enol forms, and lactam forms. So if you write it, thymine, we are having keto or lactam, and enol or lactam. Guanine we are having. Guanine is present in amino and imino structure. Guanine is present in, again, keto, lactam, and enol and lactam forms. Similarly, amino, adenine is present in, adenine is present in amino and imino. Amino, imino. Difference between amino and imino is the shifting of hydrogen from nitrogen to nitrogen. In this case, it was, in case of keto, it was, or lactam, it will be carbon to carbon or nitrogen. In this case, it is nitrogen to nitrogen, hydrogen movement will be there. So adenine, it exists in two forms, it is amino and imino. Similarly, cytosine also present in amino and imino forms. So structures, I will put it and you can see it. This is, normally we are having in DNA 4 different nitrogenous bases is present, that is thymine, guanine, cytosine and adenine. So 2 purine and 2 pyrimidine. In case of RNA, we don't have thymine. Thymine will be replaced by uracil. So in RNA we are having 4 types of nitrogenous bases: guanine, adenine, cytosine and uracil. In case of DNA we are having thymine, guanine, adenine and cytosine. Other than this nitrogenous bases which is present in the DNA, there are a number of nitrogenous bases analogues present in different forms. Like we are having hypoxanthine or xanthine. Those hypoxanthine and xanthines are the metabolites in case of uric acid metabolic processes. Other than that one, there are a number of other metabolites which is present in the plants like theobromine, caffeine, theophylline. These are different analogs which is present in the plant or tea or coffee. So that is, you can call it analogs. So this is about nitrogenous bases. We'll see how it is present in the DNA or nucleotides. Now come to ribose sugar. Ribose sugar is a 5-carbon sugar which is present in DNA, RNA. So this ribose sugar we are getting in our cells normally from HMP scent, hexose monophosphate scent, which is already we have seen it. So this is existing in the forms of, this is carbon number 1, 2, 3, 4 and CH2OH. At this place we are having H, OH, H, OH, H, OH and H. This is the difference. This is one sugar. This is called as, because at every places we are having OH is present. So this is called as ribose sugar. This the difference between, this is called as deoxyribose sugar. The difference between this one and this one at second carbon. So in case of ribose sugar we are having OH at the second place. In case of deoxyribose we are having H at the second place. So that is the difference. The numbering, when we are doing the numbering, we have seen the numbering in case of nitrogenous bases. The numbering we have done, that was 1, 2, 3, 4, 5, 6, 7, 8, 9 in case of purine, and 1, 2, 6, 3, 4, 5, 6 in case of pyrimidine. Because this sugar and the nitrogenous bases present in the same nucleotides. So how to differentiate between the carbon, first carbon of your first nitrogen or the third nitrogen or the second carbon of nitrogenous base and second carbon of ribose sugar. So to differentiate that one, the numbers of sugar we are going to put, this is called as 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. So prime does not, prime means just apostrophe on the top. This prime does not have any other significance. This is just to differentiate the numbers of sugar and the numbers of nitrogenous bases. Then in case of this one, the first carbon nitrogenous bases is going to associated with, going to form bond with this first carbon. So nitrogenous bases will form here. Here nitrogenous bases is going to bond with the first carbon. The bond which is going to form in case of purine and pyrimidine. So both purine is having carbon at present at, nitrogen is present at both the cases. This first OH will bond with the nitrogen of the nitrogenous bases. Which nitrogen will bond? In case of purine, we are having 4 nitrogen at place 1, place 3, place 7 and place 9. So in case of purine, it is nitrogen which is present at number 9. Means in the 5-membered ring, the 9th nitrogen that is going to form a bond. In case of pyrimidine, it is only 2 nitrogens are there, that is place 1 and place 3. So this is nitrogen 1. So you have to remember the two, in case of difference between the sugar, the ribose and deoxyribose have a difference at the second places. So second places in case of ribose sugar we are having OH, and second place we are having H only in case of deoxyribose. The first carbon OH is associated with the nitrogen of the purine or pyrimidine. In case of purine it is nitrogen, which is ninth nitrogen. In the case of purine it is nitrogen 1. So these two are bound through glycosidic bonds. So this is sugar. In case of sorry, so they are going to form with the glycosidic bonds. Now this CH2, if you see the structures, so we are having this, if you see one by one. Like I am going to write a structures like this is R, this is CH2OH, and this is carbon number 1, where I am going to form a bonds with nitrogen of anything, any purine or pyrimidine. If this is nitrogen number 9, so this nitrogen number 9 can exist this is one kind of structures where this is opposite side to of this sugar. This is not on the sugar, so this is called as anti, where sugars, this ring will not on just above the sugar, that is called as anti structures. If this will exist in such a manner where this will be present here, like this is, just remember this is anti structures. Second structure we are calling it syn, where this is ring is just above the sugar, so this is called as syn. So there are two different existence of, this is sugar, this is nitrogenous bases of combination of sugar and nitrogenous bases we are calling nucleoside. So nucleoside present in two structures, that is called as syn structures and anti. So if it is adenine we can call it syn adenosine, if it is anti we can call it anti-adenosine. Similarly, in case of guanine, in case of thymine, in case of cytosine, we are having syn and anti. So this is sugar. Now we will say the names. What we are calling nucleosides. In case of, if we are having, we have 5 different things. If we are having adenine, thymine, cytosine, guanine and uracil. If it is exists in case of oxy form or deoxy form, means the sugar will be oxy or deoxy. In case of ribose, in case of deoxyribose. In case of ribose we will call it adenosine. In case of deoxyribose we will call it deoxyadenosine, or more specifically we will call it 2-deoxyadenosine. Similarly, thymine we will call it, because it is not present in case of ribose, so thymine is absent here, because I have told you thymine does not present in the RNA. So ribose is present in RNA because it is ribose sugar. So thymine only present in case of DNA. So we will have 2-deoxy thymidine or thymidine. Similarly, cytosine we are having present in both the places. So we'll call it cytidine or 2-deoxycytidine. Guanine also present in both the places, so we'll have guanosine and 2-deoxyguanosine. Yeah, uracil does not present in DNA, it is absent there. Uracil is present only in case of RNA, so you will have uridine. Now this is this is the nucleoside. When nucleoside will have a phosphate, we will call it nucleotide. So nucleoside plus phosphate is called nucleotides. So if this nucleosides can link with 1 phosphate, 2 phosphate or 3 phosphate. So it can be monophosphate, it can be diphosphate, it can be triphosphate. Like if only 1 phosphate will be there, it is called as, in case of this one, adenosine monophosphate. In case of 2-deoxy, adenosine monophosphate. If 2 phosphate will be there, it will be adenosine diphosphate. 3 phosphate, it will be adenosine triphosphate. Similarly here 2-deoxy adenosine monophosphate, 2-deoxy adenosine diphosphate, 2-deoxy adenosine triphosphate. So similarly in all the cases we will have mono, di and triphosphates. Now we'll see how this is going to link together. So how nucleotide and nucleoside is going to form a bond. I'm going to draw a structure for nucleotide for DNA. This is our sugar. Because I'm talking about deoxy sugar, so I will write only hydrogen, OH, hydrogen. This is sugar and this is OH. Now I have told you this is sugar. Sugar is going to combine with nitrogenous bases. So I am going to write a nitrogenous base of adenosine. Adenine is purine, so I am going to write adenine, that is 6-amino purine is called as adenine. So this is OH and adenine is having nitrogen . 6-amino purine is called as adenine. So this is R-adenine. So this nitrogen is going to form a bond with this one. This is called as glycosidic bonds. So nitrogenous bases is associated with the sugar with the glycosidic bonds. So this is called as nucleoside. This nucleoside at 5' carbon is going to be associated with the phosphate. So this is CH2. This OH will associated with phosphate. So this is O, P, O, P, O, P. 1 double bond, 1 single bond. 1 double bond, 1 single bond. 1 double bond, 1 single bond. And here double bond. Because this is 2-deoxyribose adenosine triphosphate. This is 2-deoxyadenosine triphosphate. This is one nucleotide. So this nucleotide, we will have a number of nucleotides. So nucleotides are joined each other by this if you see how two nucleotides is going to form the DNA strands. So this one is going to join with this one of the third. So always there is a joining between the 3' OH, 3', this is 3' OH and the 5' phosphate. We'll see how this is going to form. So if you'll see this one, you can remember this one. So you can remember that in case of one nitrogenous basis, one nucleotides, nucleoside, we are having a glycosidic bonds. So if this is OH, this is CH2 and phosphate, this is nitrogenous basis, this is one, this is H. Similarly we are having H, nitrogenous basis 2, nitrogenous basis 1, H, OH, CH2, phosphate. So this is one nitrogenous base nucleotide 1, this is nucleotide 2. So I have told you this OH, 3' OH is going to combine with the 5' phosphate. So what will happen at this place? We are having at one side we are having oxygen, at one side we are having OH, which I have told you in case of O-. So this and this combine together and forms a bond like this one. Like this one. So phosphate is present between two ester groups. So this is called as like bond between like OPO. This is called as phosphodiester bond. Phosphodiester bond is going to form between 5' CH2OH of one nitrogenous basis and 3' OH of another nitrogenous basis. So this is called as phosphodiester bond. So between means two nitrogenous bases joined together in a single strands by a phosphodiester bond. In one nitrogenous basis, one nucleotides, you are having glycosidic bonds. So this process joins and multiple. Again you will have a third nucleotides like this one, 3 CH2OH plus phosphate. So this phosphate will join with this one, this is OH. So you will have a straight chain of nucleotides. So this is one strand of nucleotides. In one strand of nucleotides, at this prime, 5' phosphate is free at one end. In another ends, you will see 3' OH is free. So DNA can have one side we will have 5', and another side we will have 3'. This is called as one strands of DNA. So this is 5'3" direction. Similarly, we are having multiple different DNA. So this is 5'3" direction, 5'3" phosphodiester bonds, and we are having. Now you are knowing that DNA is basically, in our case, it is double helical structures, and that has been on the basis of Chargaff rules. Chargaff rule already I have uploaded the video. I will give you the link in the description box, where purines and the pyrimidines will have an equal sharing, equal percentage in case of double stranded DNA. So adenine is going to combine with the thymine, and cytosine is going to combine with the guanine. Always adenine forms with thymines two hydrogen bonds. That is depending on the basis of its hydrogen donor and hydrogen acceptors. I will put the pictures and you can see it how this hydrogen donor and hydrogen acceptors forms two hydrogen bonds, where in case of thymine and cytosine there are three hydrogen bonds. So three hydrogen bonds forms between the cytosine and guanine, two hydrogen bonds between adenine and thymine. That is on the basis of Chargaff rules. Then this DNA, this is one of the strands, will have another strand. So because our DNA is double helical structures, and that double helical structure was given by scientists, that was Watson, Crick's, and Rosalind Franklin also helped us. So that Watson-Crick model we will see in the next lectures, where we will explain all the structure of Watson-Crick postulates, what are the rules, possibility, and then we will talk about differences between the different forms of DNA. In case of in our cells, we are having different forms of DNA like A DNA, B DNA, Z DNA. So first we will finish Watson-Crick, and then we will see different forms of DNA. Then we will go for replication and other things. So hope you have understand the DNA basics, how nucleotides forms, and if you have liked the video, please press the like button. If you have not subscribed my channel, please subscribe it. Thank you very much. Have a nice day.